The overall goal of this procedure is to selectively target stem cell-derived teratomas in mice for evaluation of the in vivo effectiveness of small animal external beam radiation therapy.
The main advantage of external radiation beam therapy is that it is a targeted therapy for stem cell-associated teratomas that avoids the off-target adverse effects of systemically delivered therapies.
Advances in external beam radiation therapy have enabled the specific targeting of small teratomas while avoiding damage to normal tissues, making this therapy ideal for treating radiation-sensitive teratomas.
This method can not only provide insight into optimizing strategies for cell therapies, it also has the potential to become clinically applicable in human therapies.
Begin by taking a 6-well plate containing iPSCs. To induce tetratoma formation in immunodeficient animals, add 1 milliliter of recombinant cell dissociation enzyme mix per well of a 6-well plate, containing human-induced pluripotent stem cells transduced with a double fusion reporter gene for a 5-minute incubation at room temperature. At the end of the incubation, disperse the cells by pipetting and stop the enzymatic reaction by adding an equal volume of cell culture medium to each well. Pool the single-cell suspensions in a 15 milliliter conical tube for counting. After centrifugation is complete, aspirate the supernatant and resuspend the pellet in 30 microliters of Matrigel matrix solution and place the tube on ice. If utilizing double fusion reporter gene transfected cells, these double positive FACS cells should also be suspended in 30 microliters of matrix.